Good morning, everyone. In today's lecture, we will discuss about the first cranial nerve, Nervus olfactorius. We will discuss about the basic principles of the olfactory system and how it gives rise to the sense of smell. The olfactory nerve is a purely sensory cranial nerve that is one of two cranial nerves which are not associated with any parts of the brain stem. The term of olfactory comes from the term olfaction, which is really just the sense of smell. We will cover the different stages of the olfactory pathway, starting from the initial sensory transduction to the different divisions of the brain. Let's begin our discussion in the region of the nasal cavity. This is a sagittal cut where you can see the tongue, the hard palate, the soft palate with uvula, the nasal cavity, and the cribriform plate of ethmoid bone. At the roof and upper part of the nasal septum and lateral wall, we can find the olfactory epithelium, this area, with the external segments or cilia of olfactory receptor neurons. Olfactory receptor neurons are very important neurons that allow transduction chemical odorants into neural signals that eventually allow us to process of smell. The olfactory sensory neuron is considered the basically bipolar neuron structure. The cell bodies of this neuron is present in the olfactory epithelium. Olfactory epithelium has approximately 10 centimeters square. The odorant, which are chemicals which are present in the air and need to sniff it to this olfactory epithelium. Now, this olfactory epithelium has special type of receptors for these odorants, because these neurons are known as olfactory receptor neurons. The olfactory epithelium, which in addition to the olfactory sensory neuron, also made out of other types of cells. There are supporting cells, basal cells, Bowman glands, and stem cells. This is a cribriform plate of ethmoid bone. This layer is lamina propria. This layer is olfactory epithelium. And this is a mucous layer with odorants. It's clear? There are the supporting cells, also known as sustenticular cells. The function of supporting cells is to provide mechanical and physical support to the olfactory receptor cells. There are Bowman glands which visit above the lamina membrane of the lamina propria, the main function of which is to produce a mucus to the mucous layer that protects the cells. Another important function of the mucus is trapping and dissolving the odorants. Another types of olfactory epithelium cells are the basal cells, which are rest on the basal lamina of the olfactory epithelium. There are stem cells, which are responsible for a proliferation and formation of new olfactory sensory neurons and supporting cells. So, olfactory receptor neurons are one type of neurons which capable to regeneration. due to proliferation and differentiation of the stem cells. Constant division of basal cells leads to the replacement of olfactory epithelium every 2-4 weeks. Now, let's cover more detail about the structure and function of olfactory respiratory neurons. There are about 10 million of these cells in the olfactory epithelium, which are interspersed among the sustentacular or supporting cells. As I said earlier, these neurons considered unmyelinated bipolar neurons. Olfactory respiratory neurons have two functional units, one that goes in the nasal cavity and converts the odorant signals in the neural signals, and another one that is transmits the signals to the brain structures. This functional division is made possible mainly because the olfactory respiratory neurons are bipolar neurons. The cell bodies of the olfactory respiratory neurons are present in the olfactory epithelium. This is the cell bodies of the olfactory respiratory neurons. The end that is in contact with the mucous layer and with odorants is referred to the apical end and contains itself the dendrites with olfactory knobs. This is the olfactory knobs, this is a dendrite, and there is a cilia olfactoria. Each olfactory receptor neurons has a single short dendrites that extends towards the surface of the nasal cavity. At the tip of these dendrites is expanded structure called the olfactory knob or clava olfactoria. From the olfactory knob arises multiple, approximately 10 hair-like projections called the cilia. This is the cilia, which project out of olfactory epithelium and projecting into the mucous layer covering the nasal cavity, the roof of the nasal cavity. The olfactory cilia are immotile and contains the odorant receptors for the specialized odors. This membrane is showing the ciliary membrane and the presence of receptors, G proteins, enzymes, ion channels, and exchangers. This is a G protein coupled receptor. This is a G protein. There are adenyl cyclase type 3. This is a cyclic nucleotide gated ion channels. This is a calcium gated chloride channels. And there are sodium calcium exchanger or NCX and the NKCX. So, olfactory transduction begins when an odorant binds to the G protein couplet receptor. This is the odorant. The binding of the odorant will activate and change the conformation of the receptor, which in return will be able to activate the G protein. G protein has 3 subunits, G alpha, G beta, and G gamma. This G protein is normally bound to alpha GDP. When G protein couplet receptor interacts with G protein, GDP is replaced by GTP on alpha subunits. Alpha subunit separates from beta and gamma complex. The activated alpha subunit then goes on to activate the olfactory specific adenyl cyclase type 3 or simply AC3 and binds to a specific site on this enzyme. Then the active AC3 will convert ATP to cyclic AMP. The newly made cyclic AMP are then able to bind to cyclic nucleotide gated ion channels and opens them, which leads to sodium and calcium flow inside the cell. The entry of these two positively ion species contributes to first depolarization of the olfactory receptor neurons. Now, at this point, the neuron is slightly depolarized because the signal is not strong enough to cause an action potential. An action potential actually comes from the opening of calcium-gated chloride channels by the binding of this calcium. Inside olfactory neuron, the concentration of chloride is unusually high because, in opposite to most neurons, in olfactory neurons, when the chloride channels opens, the chloride flows out of cell instead of entering inside the olfactory cell. If there are a sufficient amount of sodium and calcium enter inside the cell, inside of the cell becomes more positively charged, and outside of the cell becomes negatively charged because of chloride exit from the cell, which leads to strong depolarization. Strong depolarization causing receptor potential and then action potential. And this neuron signal travels along the axon of the olfactory neuron and reaches to the olfactory bulb and eventually to the brain and occurs smell perception. These all mechanisms were about depolarization steps. So next about recovery and adaptation, which occurs after the depolarization and perception of the smell. Adaptation is the gradual decrease in sensitivity to a continuous or repeated odor stimulus, even though the odor is still present. So the smell becomes less noticeable over time. A good example of adaptation is when you enter a room with strong perfume, you will smell strongly at first. However, after a few minutes of being in that place, you will probably notice that this smell dissipates, although the same odorants that make up the smell are presumably still there. This phenomenon is explained by the process of adaptation. So next, let's see mechanisms of adaptation step by step. During continuous odor exposure, the first step is prolonged opening of calcium and natrium channel, cyclic nucleotide gated ion channels, and calcium keeps entered inside the cell, and amount of intracellular calcium increases. Second step is calcium binds to calmodulin and forms the calcium-calmodulin complex. Third step, calcium-calmodulin complex directly binds to the cyclic nucleotide gated channels and decreases the channel sensitivity to cyclic AMP. Even if cyclic AMP is present, channels opens less, which will lead to reduce the amount of sodium and calcium that enters. Calcium-calmodulin complex activates phosphodiesterase. That hydrolyzes cyclic AMP to AMP, which leads to reduce the amount of depolarization because AMP cannot open the cyclic nucleotide-gated ion channels. Calcium-calmodulin complex also activates calcium-calmodulin kinase. Calcium-calmodulin kinase 2, which leads to phosphorylation and inhibition of adenylyl cyclase type 3. As you can see, calcium has many effects in this pathway. Another component which play a crucial role in the adaptation is cyclic AMP. As I said earlier, that during the depolarization stage, the level of cyclic AMP increases. During the adaptation stage, this cyclic AMP activates the protein kinase A. Protein kinase A and G protein receptor kinase together can then phosphorylate the G protein couplet receptor, GPCR. Phosphorylation reduces the receptor activity. This phosphorylation triggers involve the B-arrestin and other adapter proteins which leads to inhibition of G protein couplet receptor. Finally, another important step which contributes to adaptation is hydrolysis of active GTP. Hydrolysis of GTP by the regulator G protein signaling 2. After hydrolysis, the alpha subunit becomes inactive and reassociates with the beta and gamma subunites, and as a result occurs reforming the inactive G protein and occurs inactivation of the signal. So, next concept about recovery. After the odor is removed, your sensitivity gradually returns to resting state. This process is called recovery. For example, while cooking, smell fades it because of adaptation. However, after going outside and returning, smell is strong again. This is due to recovery stage. Recovery starts when odor stimulus is removed. No binding of odorant to olfactory receptors. Receptor becomes inactive. And inactivation of G protein via the IGS2 regulator G protein signaling 2 with the formation of inactive GDP of the alpha subunit from the active GTP of the alpha subunit. Next, stopping activation of adenylyl cyclase type 3. As a result, adenylyl cyclase type 3 can convert AMP to cyclic AMP. Cyclic AMP returns to a basal level due to phosphodiesterase, which will convert the cyclic AMP to AMP. Inhibition and desensitization of the cyclic nucleotide-gated ion channels due to calcium-calmodulin complex stops sodium and calcium entry. Next, removal of intracellular calcium by the specific transporters. There are 3 known transporters that can be used by olfactory receptor neurons. There are NCX, NKCX, and ATP, calcium ATPase transporters. The difference between NCX and NKCX is which ions are used to remove calcium. So, the NCX The NCX, or in another word, sodium-calcium exchanger, removes calcium using the natrium gradient, the sodium gradient, sodium, and removing of the calcium. The NKCX, sodium potassium calcium exchanger, removes the calcium using both sodium and potassium gradient. Removing calcium. Calcium ATPase uses pumps for removal of calcium out of the cell, which maintains basal resting calcium level. Before we go back to the olfactory epithelium, there are few properties about olfactory respiratory neurons and odorants that I want to discuss because they are very important. The human nose has about 10 million olfactory receptors. There are approximately 400 functional types of olfactory receptors, which can bind to odorants. So, for example, this is a schematic diagram showing different types of olfactory receptors and there are the different types of odorants. So, as you can see, this odorant is able to bind to particular odorant receptor protein. On the other hand, if you see this particular odorant has a specificity for two different types of odorant receptor proteins. So, another odorant, it can bind to two other different types of receptor proteins. So you see that there are, first of all, so many different types of receptors. That is 400 receptors. However, it not means that there are only 400 odorant can bind to 400 receptors. You can see because of this different combination of binding, there will be a lot of permutation combination by which odorant can bind to multiple receptors. This combination lead to perception of different smell sensations. So, this is what is happening at the level of olfactory epithelium, that is binding of the odorant to odorant receptors, which are present on the dendrites of the olfactory sensory neurons. And there will be generation of the action potential. Now, go back to the schematic diagram of the olfactory epithelium. The basal end of the olfactory receptor neurons has axons. The axons pass through the cribriform plate, which is basically present in the ethmoid bone. And this cribriform plate actually separates the nasal cavity from the cranial cavity. So there are perforations in the cribriform plate, and there are the axons which are crossing these perforations and reaching to the cranial cavity. In the cranial cavity, there is a region just above the cribriform plate known as the olfactory bulb. The olfactory bulb has 6 different layers. There are olfactory nerve layer, glomerular layer, external plexiform layer, mitral cell layer, internal plexiform layer, and granule cell layer. First layer is olfactory nerve layer. This is considered the most external layer of the olfactory bulb. This layer contains the long axons of the olfactory respiratory neurons, which carries raw chemical odor information from the nose into the brain for the first time. Because Each axon's bundle carries only one receptor type signals. This layer already starts sorting smells type by type. Second layer is called glomerular layer, which contains spherical processing units called glomeruli, where the first brain synapse happens. This synapse are the site of connection between the incoming axons of the olfactory respiratory neurons and the dendrites of the projection neurons. In terms of projection neurons, there are two main types of cells, mitral cells and tufted cells, which are located in the two different layers. Each glomerulus is a tiny meeting point for one specific smell type. Each glomerulus is dedicated to one odor receptor type and only receives axons from olfactory sensory neurons expressing that receptor. In addition to synapses between the olfactory receptor neurons and projection neurons, each glomerulus also contains the input from periglomerular cells. Periglomerular cells, which are small round cells that sit around the outside of each glomerulus like security guards. They connect neighboring glomeruli to each other, allowing them to suppress each other. This is called lateral inhibition, which sharpens your perception of what smell actually is. So, third layer is external plexiform layer, the major signal refinement zone, which contains the tufted cell, lateral dendrites of mitral cells, and apical dendrites of the granule cells. Tufted cells are smaller than mitral cells. This is a tufted cell. Then send smell signals to the brain. Except if their lateral dendrites run through the external plexiform layer and interact with granule cell branches in the same way with mitral cells too. Apical dendrite reaches up into glomerulus into second layer, which receives direct smell input from olfactory respiratory neurons. Another structure of the external plexiform layer is lateral dendrites of mitral cells. There is a lateral. Mitral cell bodies sit in layer 4, fourth layer. However, they extend long horizontal branches, lateral dendrites, to third layer into external plexiform layer. Another structure of the external plexiform layer is the apical dendrites of granule cells. Granule cell bodies are present in layer 6, in the granule cell layer. However, they grow their branches into this layer, third layer, external plexiform layer, to meet mitral and tufted cell lateral dendrites. This is where the crucial two-way synapse happens. Mitral to granule give excitation and granule to mitral cells inhibition. Back. This feature prevents two similar smells from being confused by suppressing overlapping responses in neighboring mitral cells. So, except it normalizes smell signals. Fourth layer is mitral cell layer, which contains mitral cells. Mitral cells are large triangular neurons which have very distinctive architecture, and one apical dendrite reaches up into glomerulus, into second layer, which receives direct smell input from the olfactory sensory neurons axons. Multiple lateral dendrites spread horizontally into third layer. They receive inhibitory feedback from granule cells, allowing signals. One long basal axon exits the ball through the layers 5 and 6, joins the lateral olfactory tract, and projects directly to olfactory cortex. So fifth layer is internal plexiform layer. Internal plexiform layer, which contains mitral cell axons, tufted cell axons, and centrifugal fibers from the olfactory cortex. The axons from the mitral and mitral layer and external plexiform layer axons passes through this layer on their way out of the ball into the lateral olfactory tract. The last layer is granule cell layer, the largest and deepest layer of the olfactory bulb, which contains granule cells. This is the most numerous cells in the olfactory bulb. They haven't axons at all. Instead, they communicate entirely through their dendrites. Basal dendrites receive cortical feedback from piriform cortex. Apical dendrites reaches to third layer to form two-way synapses with mitral tufted lateral dendrites. Granule cell layer continuously receives brand new neurons formed in adulthood, allowing the smell system to adapt and lend new smells throughout of life. We are now ready to move on to what happens in the brain and to do. This is a bottom view of the brain where you can see the olfactory bulb, where the signals from the olfactory respiratory neurons integrated. The continuation of the olfactory bulb is olfactory tract. The olfactory tract is a narrow flattened band of white matter running along the inferior ventral surface of the frontal lobe. It lies within the olfactory sulcus. Fiber composition of the olfactory tract are mitral cells, tufted cells, and centrifugal fibers to olfactory bulb. As olfactory tract reaches the region of optic chiasm at the anterior perforated substance, the olfactory tract divides into three stria: lateral olfactory stria, medial olfactory stria, and intermediate olfactory stria. Each stria then sends olfactory information to different cortical areas. The destination of the lateral olfactory stria are primary olfactory cortex. The destination of the medial olfactory stria are septal nuclei, hypothalamus, and contralateral bulb. The destination of the intermediate stria are anterior perforated substance to olfactory tubercle. The primary olfactory cortex is a collective term for all cortical areas that receives direct monosynaptic input from mitral and tufted cells of the olfactory bulb. This region are sometimes collectively called the rhinencephalon, nose brain. All components of the primary olfactory cortex are anterior olfactory nucleus, olfactory tuberculus, piriform cortex, amygdala, and entorhinal cortex. Piriform cortex is an area which occupies the medial temporal lobes. Lateral olfactory stria terminus. Main function of piriform cortex is odor discrimination, identification, and pattern recognition. Piriform cortex outputs are to amygdala, which function is odor via conditioning and emotional associations. Another output to entorhinal cortex, area 28, then via perforant path to hippocampus, which function is memory. Another output of the piriform cortex is to orbitofrontal cortex via mediodorsal thalamus, which function is conscious odor identification. Hypothalamus. Another output of the piriform cortex is to hypothalamus through the amygdala, which function is feeding and reproductive behavior. Another output of the piriform cortex is to olfactory bulb through the centrifugal fibers. The main function is top-down modulation of odor processing. Another area important in the identification of odors is entorhinal cortex. Entorhinal cortex is primary olfactory cortex area and also considered which receives projections from piriform cortex, which occupies parahippocampal gyrus Brodmann area 28. Main function of entorhinal cortex is considered gateway to hippocampus. The function of hippocampus is olfactory memory consolidation. In patients with Alzheimer's disease, this region of the brain sometimes degenerates and leads to an associated loss of being able to tell which odor is smelled. Another structure of the olfactory cortex is anterior olfactory nucleus, which are located within the olfactory tract. This area is anterior olfactory nucleus. Here located anterior. The anterior olfactory nucleus is responsible for providing connections between two olfactory bulbs via the anterior commissure, because the main function of anterior olfactory nucleus is bilateral olfactory integration via the anterior commissure. Another structure of the olfactory cortex is olfactory tubercle, which is located at the anterior perforated substance, ventral striatum. The main function is reward and hedonic value of olfaction. Another structure is amygdala, which receives direct input from the olfactory bulb via the lateral olfactory stria. Except it, amygdala receives projections from the piriform cortex. Amygdala outputs are to hypothalamus, which function is autonomic and neuroendocrine via response. Another output to orbitofrontal cortex via the thalamus, which function is conscious odor perception and identification. Another output of the amygdala is to hippocampus, which function is learned odor via association. To sum up, the main function of the amygdala is emotion via responses to sense of smell. Another structure is thalamus, which is considered a secondary olfactory cortex. All other senses, vision, hearing, touch, taste, rely through the thalamus before reaching to cortex. However, olfaction projects first to the primary olfactory cortex directly and only secondarily via the piriform cortex and amygdala rely through the thalamus to reach orbitofrontal cortex. This is a fundamental anatomical distinction. The thalamus is a collection of nuclei, in particular the mediodorsal thalamic nucleus, which is considered the key olfactory relay nucleus of the thalamus, which forms the critical link between the primary olfactory cortex and secondary olfactory cortex to orbitofrontal cortex. The orbitofrontal cortex is also secondary olfactory cortex, which sits on the orbital surface of the frontal lobe and processes olfactory information from the medial-dorsal thalamic nucleus. Main function of orbitofrontal cortex is odor perception and identification, naming a smell. Except it, orbitofrontal cortex function is learning which odors predict reward or punishment. Damage to orbitofrontal cortex can cause olfactory agnosia. The patients with such disease can detect a smell, however, they cannot name it or identify the smell. Next structure is hippocampus. Hippocampus does not receive direct olfactory input from the olfactory bulb. All information to hippocampus indirectly via the entorhinal cortex, area 28, and amygdala. Hippocampal functions in olfaction are episodic olfactory memory, memory consolidation, and distinguishing between similar odorants in memory storage. Another structure is hypothalamus. The hypothalamus also does not receive direct olfactory bulb input. Olfactory signals reach to hypothalamus via the four indirect pathways. There are first medial olfactory stria via septal nuclei, second amygdala via stria terminalis of the lateral olfactory stria, third olfactory tubercle, and the last one is diagonal band of Broca. Hypothalamus is an important region for autonomic behaviors. With respect to olfaction, it mediates the involuntary reactions our body produces, such as salivation, gastric activity, and so on.